In the previous lecture, we focused on the cell cycle and mitosis. In this lecture, I'm going to talk to you about how meiosis and sexual reproduction differ from mitosis. Asexual reproduction does have benefits. When it comes to, for example, bacteria, asexual reproduction doesn't require a partner. They can make lots of copies in a short amount of time. If they've got a combination of genes that's working well, then making lots of copies of that same combination is going to result in an entire army. Unfortunately, the negative part is that because they're all exactly the same, if an environmental condition comes around that they cannot handle, it would defeat all of them at once. And that's the benefit that sexual reproduction offers. It allows for variation and new combinations to form, and in some cases, this can offer an evolutionary advantage, giving a trait that didn't exist before or a new combination of traits that helps an organism survive better than it would before. So going back to our previous lecture, the important material that's in our cells that gets passed on from parent to offspring is our DNA. DNA contains codes called genes, and genes are codes for making specific proteins in our body. So the DNA and these genes are packaged on chromosomes. Now, speaking about humans, we have 46 chromosomes. However, that is our 2N or diploid number. In other words, if we were to count how many different chromosomes we have, completely different, that code for completely different information, there would technically only be 23 different chromosomes. But since we receive one set of 23 from mom and a second set of 23 from dad, our body cells have 46 chromosomes because our chromosomes are in pairs. Now, of those 23 pairs of chromosomes, 22 are called autosomes, and one pair is specifically called sex chromosomes. They're called that because, among other things, they carry the information to make us male or female. XX results in a female, whereas XY results in a male. When you look at a karyotype, you can recognize the difference because the X chromosome is actually a lot bigger than the Y chromosome. X will look something like this, and Y is really small. That's because the X chromosome actually carries a lot more information than just making somebody female. It controls a lot of other things, such as the ability to see color and the ability of your blood to clot. The Y chromosome contains very little information other than its presence making you automatically male. So this diagram is actually showing an organism that has a 2N number or a diploid number equal to 6. And what they've done is they're showing the red representing chromosomes that originally came from mom, 1, 2, 3, and the blue representing chromosomes that originally came from dad, 1, 2, 3. However, you'll notice at the same time that these chromosomes also match each other. You have 2 of pair number 1, and 2 of pair number 2, and 2 of pair number 3. And those are called homologous pairs. So this diagram shows the sexual life cycle of a typical animal. What we have is, if we start over here, the diploid cell, that's the zygote. That grows into a multicellular organism through mitosis and development. The multicellular organism then produces sperm and egg by meiosis. Those haploid cells get fertilized and grow into a diploid organism again. So what we see is a cycle where there's a haploid portion of the cycle, fertilization, and then a diploid portion of the cycle. This is something that's common to all sexual life cycles. We have meiosis at some point, we have fertilization at some point. We have haploid, and we have diploid. Now you might assume that all organisms show the same thing, that the adult is multicellular, they make sperm and eggs, the sperm and eggs fuse, and we get the multicellular adult again. But that's actually not the case. There are actually 3 main types of sexual life cycles. The first one is what I just showed you in animals. the multicellular diploid organism exists. That's like us as adults, children, etc. Then we make haploid gametes. Those gametes fuse together and grow into a multicellular diploid organism again. The second type of life cycle is called alternation of generations. And I'm going to show you that one in a moment. And the last one is where the haploid organism is multicellular and the spores that are diploid are only single-celled. This will make more sense when I show you some pictures. So this on the left is our typical animal. We have our fertilization. This is our multicellular organism. Most of the entire life is multicellular and diploid. And then we make gametes. This is our sperm and our egg. They get fertilized and we get our multicellular organism again. If we were to draw this as far as the amount of time spent haploid and diploid, it would look like this. 2N for a majority of the life, and N for this little tiny section. In the middle, we have what we see in plants. This is what's called an alternation of generations. We actually have fertilization, and then we get our zygote, which is 2N, and this grows into a multicellular 2N organism. So multicellular, and it's diploid. But what then happens, believe it or not, is that we end up making haploid spores right here by meiosis, and we end up with a multicellular haploid organism, which then makes these little gametes that fuse together, and those grow into a multicellular diploid organism. So you could actually see the same plant, and it would look different if it was the haploid version of the plant versus the diploid version of the plant. So again, here's, for example, the same exact plant, and down here, this is the 2N or diploid multicellular one. It then produces these little spores by meiosis. Those actually grow into a multicellular haploid organism, which then makes gametes that fuse, and that grows into a multicellular diploid organism. So what we see in this one, if we go back here, this was our animal one. It spends pretty much the whole life 2N, and this little portion is N. What we would see in a plant is exactly half and half, where we would have half of the life spent diploid and half of the life cycle spent haploid. Finally, when we come over here to the fungus, over here, we see the exact opposite. The zygote, which is 2N, is actually single-celled. It immediately undergoes meiosis, and the haploid organism becomes multicellular. So if we were to draw what we would see for a fungus, it would look the opposite of us. It would be N for a majority of its life, and 2N for just this little segment. So just to clarify, this is what it looks like in a plant. Again, remember, in an animal, that's the one we're familiar with. The whole thing was 2N. Only the sperm and the egg were N, and then those fused together. This is what we see in a plant. Here's our zygote, like a fertilized egg, but that actually grows into a multicellular organism, which is what we do. But then, when meiosis happens, we end up with spores that are haploid. Those spores go through mitosis and become multicellular. So you could actually look at one of these, and it would actually be a multicellular haploid organism. It then, through mitosis, makes gametes. Those fuse together, and the zygote then goes through mitosis and grows into a diploid organism. So we end up with a multicellular diploid organism, as well as a portion of the life cycle that's a multicellular haploid organism. This is the fungus again. Here's what happens in a fungus. We have fertilization. We make a zygote. It's 2N. But that never grows into anything. It immediately undergoes meiosis to make a haploid cell, and the haploid cell grows into, if it's a mushroom for example, that haploid cell grows into the adult organism, multicellular, but it's haploid. That then produces haploid spores. When two of those fuse together, it makes this zygote, but that undergoes meiosis immediately to form the multicellular haploid organism again. The bottom line is that no matter which life cycle we're talking about, we always see at some point the chromosomes get cut in half, and at another point, the chromosomes get doubled. So there's always going to be meiosis at some point, and there's always going to be fertilization at some point. The difference is whether that grows into a multicellular organism or not. Also notice that it is possible for a haploid cell to undergo mitosis. In other words, if there's just one copy of the chromosomes, it can still make exact copies of itself. But you can only have meiosis if a cell is diploid. Okay, and now for the stages of meiosis. So just like mitosis, we're going to have the cell cycle with G1, S, and G2, but this time our reproduction portion is going to be different. There's two cell divisions, meiosis 1 and meiosis 2. We're also going to make 4 cells rather than just 2, and each daughter cell that's made is going to have half the chromosome number. So in meiosis 1, the chromosomes are going to line up side by side. Now this is important. For example, let's say that this cell has a diploid number of 4 chromosomes. In mitosis, here's what would happen. Those 4 chromosomes would line up like this, down the middle. This would split, and each cell would have gotten 4 sticks. And they would have been identical to the original cell in G1. In meiosis 1, that's not what's going to happen. These chromosomes are going to line up with their homologous partner, like this. And when they separate, we're going to end up with only 2 chromosomes in each cell. So the chromosome number has now been cut in half. We can calculate numerically how many possibilities there are if we know the base number, the n number of chromosomes. It's going to be 2 to the n. So for example, in us, since there are 23 different chromosomes, that's our haploid number, the number of possible eggs that could be made, or the number of possible sperm that could be made, would be 2 to the 23rd. If a cell only has n equal to 2, then there's only 4 possible combinations. If n was 5, there would be 2 to the 5th or 32 combinations, etc. Alright, so the diagram at the right shows what we were left with at the end of meiosis 1. So we went from 1 cell with 4 chromosomes, in this case, to 2 cells, and each one only has 2 chromosomes. In meiosis 2, those chromosomes would line up in a straight line, just like in mitosis. And now what would happen is that each of those chromosomes would split at the chromatid, at the centromere, and we would end up with a total of 4 cells, each one with 2 chromosomes. So it would look something like this. 1, 2, 3, 4. Notice that the chromosome number did not get cut in half a second time. If we were to go back to the original cell, that original cell before S would have had 4 sticks. Now, at the end, instead of 4 sticks, we've got 2 sticks. Now, the other thing is that this is what happens in spermatogenesis. Each of these would become a sperm. But in oogenesis, we only get 1 egg, and that's because the cytoplasm doesn't divide evenly. So in meiosis 1, we end up with 1 egg with all the cytoplasm, and then in meiosis 2, again, 1 egg with all the cytoplasm. And so we only end up with 1 viable egg for every pre-egg cell rather than 4. this illustrates exactly what I just said. This is sort of our pre-egg and pre-sperm cell. After S, they look like this. Those would line up side by side. And in meiosis 1, right here, meiosis 1, we would get one copy of each chromosome. And it would be completely random which one of the pair it was. They happen to show the two red ones ending up in this cell. It could have been a red and a blue, just like happened on the left. This is our first polar body, and then our second polar body. And then these four become sperm, but this one only becomes one egg. Okay, crossing over is really important because not only are there 2 to the n number of possibilities of what we can get in an egg or a sperm, this is actually further increased because what happens is during meiosis 1, when the chromosomes line up, they don't technically just line up next to each other like little soldiers. What really happens is they sort of wrap around each other like this and pieces of information cross. So when this happens, what we can imagine is now when these chromosomes get ready to split, let's just say that this little piece here switched places with the other little piece. So at the end of meiosis 2, if we were to look, we would actually see that all 4 of these cells are completely unique because of the crossing over that happened between this chromatid and this chromatid. Now, whereas in mitosis the chromatids on a single chromosome were identical to each other, they're not identical to each other anymore. When it comes to meiosis, each chromatid may look somewhat different from its partner once crossing overs happened. This diagram shows the same thing. If we had a pair of chromosomes, crossing over occurs right here. After crossing over, some pieces have switched places. We have meiosis 1 and then meiosis 2, and notice that all 4, if these were sperm, all 4 of these are actually somewhat different from one another. Okay, so I want to summarize a couple of important concepts in these last couple of slides. So let's say we have a cell with a 2 and number of 4. Now what I've done is, this is going to be chromosome pair number 1, and I made them slightly different colors just so we could keep track of them. And here, this is going to be chromosome pair number 2. So these are homologous pairs. Well, the first thing I want to show you is what is meant by independent assortment. Remember that our rule for independent assortment is that the number of possible outcomes was 2 to the n. So in this case, n is 2. There's only 2 pairs of chromosomes. So that means there's 2 squared, or 4 possibilities. Well, let me show you what they mean by 4 possibilities. So here's what we're saying. When these chromosomes line up, they could line up with the light red one on the left or the light red one on the right. And depending on how these line up and how these line up, there's actually going to be 4 possible outcomes when meiosis happens. So here's what happens. Our chromosomes could either line up, like we said, with this light red one on the left and the dark red one on the right, or opposite. Depending on how they line up, at the end of meiosis 2, there's actually 4 possible outcomes. We could get the light red and the light green chromosome together, the dark red and the dark green chromosome together, the dark red and the light green, or the light red and the dark green. That's what we mean when we say independent assortment. And so, in this case, 2 squared, or 4 possible outcomes. In us, if we wanted to show all the possible types of eggs that could be made, in us, since there's 23 pairs of chromosomes, we would be drawing 2 to the 23rd different possible outcomes. And the last thing I want to illustrate for you is how crossing over would affect the outcome in this cell. So we're going to start with our same cell, but now I'm going to have a couple of small parts of these chromosomes cross over with their partner and then see how the outcome comes out. Okay, so here's our cell after crossing overs happened. Now, assuming the chromosomes line up this way, because again, we still have independent assortment, but we're going to pretend like this is the lineup. And now this is going to go through meiosis 1. So let's take a look at what happens in meiosis 1. So here we are at the end of meiosis 1. And you'll notice that the chromosome number has been cut in half because the partners split. Now let's see what happens when we go through meiosis 2. Alright, and here are our 4 cells at the end. So you'll notice that each of these is unique in comparison to all the others. Technically, if there was no crossing over, these 2 cells would be identical to each other because all that happens in meiosis 2 is that these chromatids split from one another. But because of crossing over, each one of these is different, and then if each one of these becomes a sperm, which then fertilizes an egg, we've now got a completely new set of chromosomes, not just because it was independent assortment or random segregation of the reds and the greens, but also because of crossing over.